bringing you the essence of great books from around the world. Hi, I'm Sally, and this is Sally's Book Talk. Today, we'll be discussing the book The Vaccine Race, subtitled The Human Cost of Fighting Disease. The COVID-19 pandemic that erupted in late 2019 has totally transformed our lives, leading to hundreds of millions of infections and millions of deaths globally.
Meanwhile, humanity has been actively battling the ever-mutating COVID-19 virus, and vaccination is the key weapon.
This is thanks to the groundwork built by generations of scientists in vaccine research. That's how COVID-19 vaccines were developed rapidly. History records that the earliest vaccines emerged in 16th century China, using skin scraps from smallpox survivors to prevent the virus. Yet, in the 1960s, vaccine research faced numerous difficulties, not just scientific challenges, but also ethical dilemmas, government oversight, and intellectual property disputes. This book recounts how scientists, led by figures like Leonard and Hayflick, raced to develop vaccines amid outbreaks in that time. The author, Meredith Wadman, is a reporter for Science and Nature and writes biology and biomedical columns for outlets like the Wall Street Journal, the New York Times, and the Washington Post. I'm looking really bad. The book draws on first-hand interviews and data gathered by Miss Wadman as a reporter for Nature. Since its release, the book has earned rave reviews from Science, Nature, and the Washington Post. We'll break down this book into 3 parts. First, how Hayflick found the key breakthrough with Wisconsin 38 cells in vaccine research. Second, what impressive successes did Wisconsin 38 cells achieve in the vaccine competition? Third, what kind of troubles did Wisconsin 38 cells cause for Hayflick? First up, let's explore the scientific backdrop of the 1960s when Hayflick hit upon his vaccine breakthrough. Creating a virus vaccine requires 5 steps. Step 1, find the disease-causing virus. Step 2, grow the virus, because you need enough pure virus to make plenty of vaccine. Step 3, weaken or kill the virus. Step 4, test for effectiveness and safety in animals. Lastly, run clinical trials in humans to show it's safe and works, so the vaccine gets approved. These 5 steps might seem simple, but in the 1960s, each one presented its own unique challenges. First, virology was still in its infancy. Scientists had extremely limited knowledge about viruses, and most viruses hadn't even been isolated yet. Second, there was no established method for amplifying viruses successfully. And unlike bacteria that multiply rapidly in sugar solutions, viruses require living cells to replicate. But scientists didn't know which cells to use for which viruses, or even how to culture monkey kidney cells long-term. The only accepted cells at the time came from freshly euthanized monkeys, yet not all viruses grew in them. Even more troubling, scientists discovered monkey kidney cells harbored a cancer-causing virus.
If vaccines were made using these cells, SV40 could contaminate them, potentially giving recipients cancer, a completely unacceptable risk.
This left scientists effectively without any usable cells. Thirdly, should viruses be inactivated or attenuated? If attenuated, how to achieve it? The scientific community was deeply divided. Finally, clinical vaccine research lacked strict regulations. The book documents ethically gray practices. Orphans and homeless people from vulnerable populations became test subjects. Thus, 1960s vaccine development faced not just scientific hurdles but profound ethical and regulatory challenges. The most critical problem was finding universal, safe cells for efficient viral amplification. The solution came from the book's protagonist, Leonard Hayflick, who discovered Wisconsin 38 cells using human fetal tissue. These cells could culture all known human viruses and theoretically provide unlimited vaccine material. He also discovered the Hayflick limit, where normal cells die after about 50 divisions, founding the field of human aging research.
Yet, Dr. Hayflick achieved this almost serendipitously, and that's where we'll begin.
Let's delve into the details. Leonard Hayflick was born in the United States in 1928 to a Jewish immigrant family. They weren't well off, but managed to get by. Leonard was very bright and showed an early passion for science. At age 18, he was accepted into the prestigious University of Pennsylvania, majoring in microbiology and receiving systematic training in bacterial culture. He then pursued his doctoral degree. His research focused on culturing mycoplasma. Mycoplasma are microorganisms between viruses and bacteria in size. Like viruses, they must live inside host cells to survive, making them equally challenging to culture as viruses. When Leonard earned his doctorate in 1956, he had become one of the era's top microbial culture experts, laying a solid foundation for his later achievements. Shortly after graduation, Leonard joined the Weister Institute at his alma mater, the University of Pennsylvania, to work on cell culture. Founded in 1892, the Weister Institute holds a prestigious and pivotal position in American biomedical history. He was recruited by Dr. Hilary Koprowski, then the new director of the Weister Institute. Dr. Koprowski was a renowned virologist, ambitious and radically transforming the institute into a premier center for virology research. Hilary valued Leonard primarily because he saw him as a competent cell culture technician who could support the institute's virologists. Hilary offered Leonard excellent terms, a newly renovated large laboratory, and freedom to pursue his own research outside core duties. Naturally, Leonard wasn't satisfied with just culturing cells for others. He aspired to conduct independent scientific research. One of his research projects investigated whether viruses could cause cancer in humans. This research direction wasn't particularly novel. At the time, virologists generally believed that if viruses caused cancer, they must be detectable within cancer cells. But as a cell culture expert, Leonard held a different perspective. He reasoned that hunting for viruses inside cancer cells was like finding a needle in a haystack. If viruses were present, they'd naturally release into the culture medium. Using that medium to grow normal cells would then infect and transform them into cancer cells. This approach is logically rigorous and the experimental design is straightforward. Leonard got straight to work. He quickly obtained cancer cells from the University of Pennsylvania Hospital, and culturing them was a breeze for him. Next, when it came to culturing normal cells, Leonard paused. As an experienced microbiologist, he knew bacteria and viruses are everywhere, and any normal adult tissue cells could be contaminated by microbes. If he used seemingly healthy normal human cells for the experiment, the conclusions might be unclear. Is it from unknown viruses in the culture medium or latent microbes in the human body? Leonard thought it over. To ensure definitive conclusions, he had to use fetal cells, as they're widely accepted as uncontaminated. Using fetal cells for science was a wild idea. But Leonard's boss, Hilary Koprowski, director of the Weister Institute, fully supported him. Hilary used personal connections to get lung tissue from aborted fetuses at a hospital and gave it to Leonard. With everything set, Leonard soon identified fetal lung fibroblasts as ideal for lab culture and decided to use them for his study. But problems cropped up fast. Leonard found his cultured cells degenerated and died after proliferating for a while. But at first he thought this must be due to cell contamination. However, after analyzing them one by one, he saw it wasn't the case, which deeply puzzled Leonard. If it wasn't an external cause, then it had to be something internal to the cells. Leonard made a bold guess. Could it be that the cells are aging and dying? To prove this, he designed a clever experiment. He mixed two types of cells together to culture them. One type was from a male fetus, which had divided and proliferated 49 times. The other was from a female fetus, which had divided 25 times. Since male cells have X and Y chromosomes, and females have two X chromosomes, they're easy to distinguish under a microscope. If cell death was due to external factors, both types of cells should die. If it was natural death, the male cells with more divisions would likely die off, leaving only the female cells alive. After 2 months of culture, Leonard found the cells thriving, but only female cells remained. The truth was clear. No contamination, the cells died naturally. This was an explosive discovery. At that time, the scientific community widely believed cell cultures could be immortal. Leonard wasn't careless. He meticulously verified his findings multiple times with consistent results.
Fetal cells could divide and proliferate about 50 times before deteriorating and dying.
In other words, normal cells have a limited lifespan. They're not immortal. Moreover, Leonard discovered freezing cells didn't affect their lifespan. Cells that had divided 9 times could be frozen, thawed months later, and still divide about 41 more times. This meant cells could be cryopreserved and revived. Since cell division grows exponentially, freezing sufficient fetal cells early could yield vast quantities. Theoretically, within a cell's normal lifespan, astronomical numbers of cells could be obtained for vaccine development. And
In short, Leonard now had an endless cell bank. He abandoned his virus cancer research entirely.
he turned to vaccine research, testing these fetal cells against every available virus, and all could infect them. Even the notoriously finicky varicella virus worked, meaning fetal cells could culture any known virus. In 1961, Leonard published his findings in Experimental Cell Research, causing an immediate sensation. His paper not only debunked cell immortality and introduced the Hayflick limit of cellular aging, but also proposed a revolutionary universal cell culture method for viruses. Virologists were stunned. All wanted his cells for research, vaccine firms for development. The paper even drew U.S. government attention. The National Cancer Institute signed a contract with Worcester, guaranteeing at least $120,000 annually, starting in 1962. This massive contract accounted for 10% of Worcester Institute's annual revenue at the time. Director Hillary allocated $8,000 to renovate Leonard's lab. Though fruitful, this was only exploratory work. To better develop vaccines, Leonard set strict rules on fetal sources. Family history had to be crystal clear, with no genetic diseases or cancer in parents or ancestors, ensuring zero risk of cancer, genetic issues, or infections in the fetus. Swedish virologist Svengard was fascinated by Leonard's work and managed to provide him with a compliant fetal stomach sample. Leonard swiftly prepared over 800 vials. Each ampoule held about 3 to 4 million cells, frozen in a liquid nitrogen tank in the lab basement. Leonard gave this near-perfect cell line a distinctive name, or WI-38. Even today, some vaccines are still produced using it, and it remains widely active. In biological research, the Hayflick limit's impact was huge. It earned the 2009 Nobel Prize in Medicine for 3 scientists who explained its biology. So, how do we use Wisconsin 38 cells for vaccines? Part 2. Let's discuss what impressive successes Wisconsin 38 cells achieved in the vaccine competition. As the proverb says, those near the water tower get the moon first. WI-38 cells, being a major breakthrough for the Worcester Institute, naturally benefited its scientists first. We'll highlight Director Hillary Kaprowski's work on polio and rabies vaccines and Dr. Stanley Plotkin's rubella vaccine story. The first use of Wisconsin 38 cells was for the polio vaccine. Back in 1955, American scientist Salk developed the polio vaccine, which dramatically reduced polio cases. It was an inactivated vaccine made from rhesus monkey kidney cells, but had limited efficiency, needed multiple shots, and didn't build enough antibodies in the throat or gut. And so virologists of that era dismissed Salk. They believed attenuated virus vaccines could improve prevention. This included Hilary Laird, Cox, and Albert Sabin from the University of Cincinnati. These top scientists didn't just oppose Salk academically, they each raced to develop oral vaccines, aiming to win U.S. government approval first. All 3 were still using monkey kidney cells at that time. However, Sabin's oral vaccine development started earlier and found success through unconventional promotion in the Soviet Union, reaching over 8 million people, giving him a dominant edge. Thus, in 1960, the U.S. Public Health Service declared Sabin's oral vaccine safer and approved it for market. After this market defeat, Hillary aggressively attacked the SV40 virus risk in monkey kidney cells. advocating for Leonard's WI-38 cells to improve vaccine safety. Yet Roderick Murray, then head of the NIH's Division of Biological Standards, rejected this. Murray was a hepatitis B expert. He understood the unpredictability of viral infections all too well.
Public concern over SV40 in monkey kidney cells grew.
This put immense pressure on Murray. But were human cells truly safer? What if cultured human cells were already infected, or if cells from different populations carried unknown viruses? Murray tossed this dilemma to an expert committee. After a year, committee experts published doubts about Leonard in the top journal Science. Murray then rejected Wisconsin 38 cells. In stark contrast, the World Health Organization praised Leonard highly. WHO argued that even with unknown infection or cancer risks, human cells were preferable to animal cells, as some viruses coexist harmoniously with their hosts. Animal viruses entering humans are easily rejected by our immune systems. Soon, Sweden, Switzerland, and Croatia began developing human cell vaccines, achieving promising results. Yugoslavia even approved WI-38-based polio and rubella vaccines in 1967 and 1968, hugely encouraging Leonard. The second case for Wisconsin 38 cells was rabies vaccine. Rabies was highly concerning in the 1960s, yet vaccines still used 1885-era methods. French microbiologist Pasteur's level. Inactivated vaccines made from dried brains or spinal cords of rabies-infected rabbits. Due to outdated production methods, this vaccine contained large amounts of rabbit brain tissue, frequently causing severe allergic reactions that led to paralysis, coma, or death in recipients. Samarol, in 1958, the American company Lilly attempted a rabies vaccine using duck embryo cells. However, it proved less effective than the rabbit brain vaccine. Hillary resolved to develop a new generation rabies vaccine using Wisconsin 38 cells. The rabies virus thrived in Wisconsin 38 cells, easily yielding high-quality inactivated vaccines. Hillary patented this and licensed multiple companies through the Wistar Institute for Production and Sales, including the Murillo Institute in Lyon, France, Behring Works in Marburg, Germany, and Wyeth Laboratories in Philadelphia, USA. Later, Hillary collaborated with Iran's Pasteur Institute, proving the vaccine protected people bitten by rabid dogs. Based on these stunning results, France quickly approved Murray's vaccine. The U.S. government soon approved Wyeth's version 2, and Lilly's duck embryo vaccine vanished from the market. The third case for Wisconsin 38 cells involves the rubella vaccine. Outbreaks occurred cyclically, every 6 to 9 years in the U.S. Each rubella outbreak brought disaster to families and society. Pregnant women infected with rubella could have babies born with cataracts, glaucoma, heart defects, even if they survived. In the 1960s, scientists knew little about rubella virus, causing widespread public and government alarm.
Around 1970, another new rubella outbreak struck.
Rubella vaccine research became critically urgent. Young Wistar virologist Dr. Stanley Plotkin planned to enter this vaccine race using Wisconsin 38 cells. Stanley was born in 1932, 4 years younger than Leonard. As a former pediatrician, he deeply understood the agony rubella caused expected parents. After systematic virology training at Wistar, he committed to rubella vaccine development. His competitors included Merck & Co. and scientists from the U.S. Division of Biological Standards, regulators themselves, showing government concern. Despite two powerful rivals, Stanley believed Wisconsin 38 cells were his greatest advantage.
Merck's vaccine was first cultured in African green monkey kidney cells for 19 generations, then attenuated in duck embryo cells.
Merck named it the Benoit vaccine. The Division of Biological Standards passaged it 77 times in African green monkey kidney cells, naming it HPV-77, but WI-38 cells didn't disappoint Stanley. WI-38 cultured the rubella virus excellently. It quickly passed tests for bacteria, fungi, and mycoplasma, then proved safe in mice, guinea pigs, and rabbits. Next came human trials, but Stanley had limited resources. He used an orphanage for 3 trials with 65 orphans, but the sample was small, so he turned to a mental institution with nearly 1,000 intellectually disabled residents. After 4 studies, Stanley got positive human trial results, compiled them, and submitted to the American Journal of Epidemiology. Yet Stanley's disadvantages were stark. Merck had corporate funds. The division was the regulator itself with funding. Stanley had nothing. Unsurprisingly, the division's HPV-77 vaccine won approval. Soon after, attenuated vaccines using dog kidney cells and rabbit kidney cells also hit the market. The previously forecasted rubella outbreak year, 1970 arrived, but no outbreak occurred. The epidemic seemed contained. But the dog kidney vaccine was soon found to cause arthritis and joint pain in women and children, leading to its withdrawal. Moreover, while these vaccines produced decent antibody levels, they couldn't effectively prevent rubella reinfection. Researchers compared the WI-38 vaccine against others, including HPV-77. Results showed Wisconsin 38 better mimicked natural rubella infection, yielding higher antibodies and lower reinfection rates. Finally, the WI-38 vaccine was approved for the U.S. market in September 1978. Today, the WI-38 vaccine is America's sole rubella vaccine and used globally. Lastly, let's discuss the troubles Leonard Heiflich faced. As vaccine successes grew, Wisconsin 38's fame, trouble followed Leonard. First came the ethical issues. People started noticing this problem. Leonard's research constantly involved seeking aborted fetuses from hospitals to obtain tissues. Otherwise, he used personal connections to access orphanages, mental institutions, or women's prisons, recruiting vulnerable groups for human trials. These approaches raised huge ethical concerns. Before the 1960s, abortion was illegal in the U.S. Yet despite widespread abortions, using fetal tissue for research occupied a legal gray area. In 1972, a pamphlet, The Women's Bible and Abortion, mentioned Leonard and his vaccines with fictionalized descriptions. It claimed Leonard used cruel methods to get fetal cells for experiments, stating, "Your child's next vaccine might come from a brutally murdered baby." The pamphlet sparked a flood of angry letters. Leonard hired Stanford's legal counsel to issue a stern warning before it subsided.
In 1973, NASA selected Wisconsin 38 cells for space experiments on survival in zero gravity.
This drew protests from anti-abortion groups. They argued Wisconsin 38 cells came from aborted fetuses, deeming the experiment unethical, forcing Leonard to publicly explain again. He stated the cells came from legally obtained fetal lung tissue in Sweden a decade prior, but critics remained relentless. Leonard's second headache was ownership of the WI-38 cells. Though the U.S. government funded Wister for Leonard's discovery, they demanded the cells become government property post-contract, a view Leonard rejected. Wisconsin 38 cells were his life's work. He believed he owned them. So when the NIH came post-contract to handle them, Leonard, furious, secretly moved all cells while unnoticed, left Wister, and became a professor at Stanford. Then, in 1971, he opened a bank account called the Cell Culture Fund. For 3 years, he charged fellow researchers for preparing and mailing Wisconsin 38 cells and kept raising the fees. In just 3 years, the account balance soared to $13,000. But Leonard claimed that since cell ownership remained disputed with the government, these fees were solely for legal battles. In reality, though, Leonard wasn't satisfied just selling to research institutions. He and his wife formally registered Cell Associates, Inc. and opened a corporate bank account. He proactively reached out to industry buyers like Pfizer and Merck, even signing a $1 million contract with Merck. Within 14 months of founding, the company earned $47,000, equivalent to $200,000 in 2016 dollars. And such pricing was considered exorbitant profiteering at the time. In the 1960s, most scientists felt a salary enabling modest, unpretentious living was sufficient. True scientists didn't seek more, believing wealth would distract from their beloved work. Leonard clearly overstepped these bounds, inviting more controversy. In May 1975, the NIH planned to appoint Leonard as director of the National Institute on Aging. But first, they sent an investigative team to his Stanford lab. The team combed through every corner of Leonard's laboratory. Then, in March 1976, the New York Times published their report declaring government-owned cell samples were illegally sold, publicly condemning Leonard's profiteering. And Science magazine followed with the rise and fall of Hayflick's tragic cell line, fueling widespread criticism.
Leonard was forced to resign from Stanford.
But Leonard didn't surrender. He hired an intellectual property lawyer and fought through appeals for 5 grueling years. During this period, he had no income, even meeting unemployment benefits to support his family. Though he later found lab work, the NIH blacklisted him, cutting all research funding. Fortunately, Leonard caught a break. By the late 1980s, with the U.S. economy struggling and politicians seeking change, Congress passed the Bayh-Dole Act as government-funded research rarely reached commercialization on fire.
The act stated that universities, nonprofits, and small businesses should retain rights to their inventions to promote commercialization, with inventors entitled to royalties.
Demonstration. Signed by President Carter, universities established tech transfer offices, legitimizing scientists' profits and fueling entrepreneurship and venture capital. In this climate, the NIH no longer wished to feud with Leonard. In September 1981, they settled. Both parties gained partial ownership of the cells. The government let Leonard keep Cell Associates' profits. His lawyer finally got paid. But by then, Leonard was over 50, a scientist past his prime research years. As of our 2022 broadcast, the 94-year-old Leonard lives in California, writing memoirs and advising scientifically. As former Merck vaccine chief Alan Shaw said, "Developing vaccines may be the most fulfilling work. You see diseases disappear, because succeeding with one vaccine eradicates a disease." Leonard's legacy is immense. He created safer human cell-based vaccines, discovered the Hayflick limit, and pioneered aging research. Human cell vaccines made diseases like chickenpox and shingles nearly vanish. Few know this began with Wisconsin 38 from an unnamed mother's fetus in Sweden half a century ago. That concludes our reading of the vaccine race. Reading notes are below the audio. Until next time.